Okay, thank you everyone for joining us. I think we have people still joining, but we will start on time. And today we have the Open Box Science seminar, joint force between our typical neuroscience seminar and omics seminar. And neuroscience is typically on Thursday, omics typically on Tuesday. Welcome to those of you that come back. And as many of you know, and for newcomers, we are an NGO dedicated to make science open and available to everyone. And today we have a great honor of Joe from University of Florida as a speaker, and we also have Celeste Karch from Washington University as a moderator. So to quickly introduce Celeste, Celeste is Associate Professor of Psychiatry at Washington University in St. Louis, and she's an expert on mechanisms underlying tauopathies. I think as you will see from her moderating, her research goal is really trying to understand all the mechanisms, underlying the tauopathies like Alzheimer's disease, frontotemporal dementia, and her lab has used many different kind of functional models like primary cell lines, cultures, and induced pluripotent stem cells with neurons and astrocytes to dissect these important mechanisms that can lead us to treatment of these diseases. So with that, I'll let Celeste take over as the moderator for today's Open Box Science seminar, and she'll introduce Joe and also moderate the talk. Thank you, Celeste.
Great, thank you so much. So it's my pleasure really to introduce Dr. Joe Abisambra, and he, as you can see, is an associate professor of neuroscience and has served as the Assistant Dean of Diversity and Health and Equity at the University of Florida College of Medicine. He immigrated to the United States from Columbia, where he then completed a PhD and postdoctoral fellowship at the University of South Florida. He then went on to establish his independent lab at the University of Kentucky, and after that was recruited to join the faculty at the University of Florida. I've long followed his work on the intersection of tau and ER dysfunction and the unfolded protein response. And so I'm really excited to hear more about that today. And his most interesting non-professional achievement is that he's heroically a father of 4, all under 9 years old, during the pandemic, and delivered his second child in the car. So you can see that he's really willing to do anything. So with that, I'm happy to hear more about this incredible work on tauopathies.
Thank you so much, Kwan, for the invitation. So that's for the kind introduction. And I'm happy to discuss delivering babies in cars at any point after the seminar, if interested. So I selected a pretty dramatic title for 2 reasons. Number one, to kind of give an impact on translation, nice little play on words, but also to honor my compatriot, Gabriel Garcia Marquez, who obtained his Nobel Prize for, and I'd love to hear in the chat, who can tell me which book gave him the Nobel Prize? Zoom, you always have to give an extra 10 seconds. I would love to hear more. Nobody. It's hard.
Love in the Time of Cholera.
Close, but not quite. Good guess. Nobody? Okay, how about 100 Years of Solitude? And I'll just say that to me, after watching Encanto 5,000 times with all my 4 kids, it kind of, Macondo, the town in 100 Years of Solitude, is definitely similar to the Valley of Encanto. But anyway, this is Of Love and Other Demons, which is the title of the book, and kind of draw that parallel of tau and other demons. And so let's get started with what dementia is. And I'll just highlight, back to literature, 2 of the probably best described characters with dementia, one of them being Don Quixote, very extensive clinical history, and also King Lear are around the same time. They show changes in personality, social behavior, language ability, difficulties thinking, poor coordination at times, and psychiatric symptoms. Now I focus on tauopathies, which is the most common form of dementias. And here's a list that I've compiled of about 22 different tauopathies. The most common tauopathy is Alzheimer's disease, and here you can see some of the pathological signs of this disease as it pertains to tau aggregation. So here's cortical basal degeneration, progressive supranuclear palsy, and chronic traumatic encephalopathy. All of this abnormal staining, these aggregates correspond to tau. And you can see that these aggregates build inside different cell types. For example, in Alzheimer's, it's restricted to neurons primarily into forming these tangles, these flame-like structures. But the distribution throughout the brain is different depending on the disease, and cell types may differ, and certainly they have different shapes and different presentations. The objective of our lab has really tried to focus on what the mechanisms are driving tauopathy and how that may link to brain dysfunction. We're focusing on molecular mechanisms not only to understand the disease, but also to understand novel therapeutic targets, maybe identify those and design new strategies to treat tauopathies. With these molecular mechanisms, we may also expand this knowledge to understand broad brain function. Many questions still remain on how our brains work, and maybe at the cellular level and molecular level, we may be able to find important information here. And these are going down to fundamental processes governing brain function and memory. And to do this, we focused on tau. Here is one of the first papers described by Mark Kirchner at the end of the 1970s, showing the tau associated with tubulin. And since then, it was dubbed a microtubule stabilizing protein, at least because in its association with tubulin, and tubulin being such an abundant protein in the brain, it made sense that it would have an important function in the brain. Tau is a fascinating protein, especially in how it may present itself and be regulated. For example, tau is alternatively spliced. You can see here representing these boxes, we have amino terminal splice variants. These two exons can be cut out here. Exon 10 can also be spliced out. And it is post-translationally regulated as well. Here we have many examples. Most of the field will have focus. If you're new to the field of Alzheimer's and tauopathies, you'll find that tau is very much established as a hyperphosphorylated or phosphorylated protein. But I also want to point out that there are sites that are glycosylated, nitrated, ubiquitinated, acetylated, methylated, and certainly cleaved. And many of these are associated with both protection in some cases, but primarily associated with disease. So to this point, I told you how much of a bad guy tau is, but it actually has a very important function in the cell. So here we have a neuron, and it has these extensions that make it quite a beautiful cell. But these extensions also present challenges. For example, if something needs to be transported from one side of the cell to the other, the challenge will be building an avenue for, say, as an example, a mitochondrion to be transported from one side of the cell to the other. These roads are microtubules, and as I mentioned, tau has affinity for microtubules, and it's described as a microtubule stabilizing protein. So once this road is built, motor proteins can help transport this cargo from one side of the cell to the other. Since the neuronal environment is quite busy, many of these microtubules are dynamic, and they can now, they need to be disassembled in order to provide more room for different processes to take place. And for that, tau is phosphorylated, facilitating the disassembly of that microtubule. Tau, again, biochemically is fascinating, and it has a region that is intrinsically disordered. That means that it doesn't quite have a stable conformation. So for reasons that are not quite clear, tau adopts a conformation that is soluble, and also in its unstable conformation, it can have pathological properties, such as promote aggregation of other proteins, maybe template them, maybe spread through other cells. What we do know for a fact is that pathologically, the brain of an individual with Alzheimer's disease will show neurons that have hyperphosphorylated tau that self-aggregates surrounding the nucleus into these tangles. But it doesn't aggregate only with itself. it has this entire interactome, and the tau interactome was described. They're just wonderful papers by the Mandelkow group, both by Eva Maria and Eckhart writing this review. Also folks in Tom Wisniewski's lab more recently, and earlier this year, Li Gan's lab showed a powerful paper with the tau interactome in cells that are undergoing depolarization. So tau not only associates with microtubules, it interacts with many other proteins. And this phenomenon is creating this emerging concept that tau has other functions beyond associating and stabilizing microtubules. Some of the work in defining the interactome was performed in my lab while I was doing my postdoc. We focused primarily on its association with the endoplasmic reticulum. And we found that tau associates with many ER-associated proteins. And the ER is essential for cellular function, such an important organelle because it connects with the nucleus by helping with translation and offering that optimal environment for proteins then to fold properly and be trafficked both to the Golgi apparatus and or the plasma membrane. If proteins don't fold properly, the ER can connect with a proteasome or autophagy to shuttle those proteins for degradation. When the ER undergoes stress, it has its own adaptive mechanism called the unfolded protein response. It connects with mitochondria through mitochondrial-associated membrane proteins, and it regulates calcium homeostasis. It also plays an important role in lipid and carbohydrate metabolism. The foundation of the work that we did in 2013 was to identify that tau interacts with these proteins. And so it leads to the idea, and we found that some of these processes are then altered. It induces ER stress. And so we didn't quite know how, which processes would be pathological as a consequence of this interaction. Some of the early stuff that we did in my lab in Kentucky was to corroborate that tau indeed associated with the endoplasmic reticulum proteins, especially pathological tau species. Here we have a human Alzheimer's brain co-immunofluorescently stained for PHF1, which is a marker of late-stage pathology, phosphoserine 396404, and calnexin, which is an endoplasmic reticulum protein on the outside of the endoplasmic reticulum membrane. And you can see that there's partial colocalization of both PHF1 and calnexin, suggesting that tau associates, and once again corroborating that it associates with ER, the outside of the ER. So to study which proteins associate with tau, we performed this very simple experiment where we had human brains with Alzheimer's or controls, and we performed a subcellular fractionation where we pulled down and enriched for microsomal proteins. And from here, we did a co-immunoprecipitation where we pulled down actin or tau from Alzheimer's or control brains, ran them on a gel, and just did our just standard liquid chromatography tandem mass spec experiments. And we did this in 2 different papers with 2 different antibodies, one that identifies the middle region of tau and one the carboxy terminal region. And that has an important reason. It's because, to our surprise, a large majority of the proteins that are associated with tau in the brain corresponded to those that played a role in translation of RNA into new proteins. So that using this C-terminal antibody of tau, we were able to ensure that it wasn't nascently translated tau. It was full mature protein associating with these other factors involved in translation. And here in this panel, I'm showing how RNA binding proteins in Alzheimer's disease were associated with tau much more robustly, and they were different compared to those in the non-Alzheimer's brains. The association of tau with ribosomes had been described over 30 years ago, in 1987, by Skip Binder's lab. But the consequences of this interaction, although it had been further studied over the past 30 years, wasn't established until we did some work in 2016. And I'll describe the results of this paper showing that tau reduced ribosomal function. And we did the study using 3 in vitro models: cell-free assay, our immortalized and stably transfected inducible HEK cell line, where we add tetracycline and cells express tau, and then finally primary neurons from tau transgenic mice. The hypothesis was that under normal conditions, tau associates with ribosomes, which we observed in some of our studies and had been described earlier, but this association would be much more robust under pathological conditions, and that would reduce the amount of new proteins that were synthesized. So our first experiment was going back in a rigorous manner to the human brains and confirm that this association with ribosomes was real. And so for this, we did, again, our human Alzheimer's brain and control subcellular fractionation, where we compared our fraction with all the entire cell lysate, and a fraction that was devoid of ribosomes and one that was enriched with ribosomes and ribosomal proteins, of course, and associated factors. And so here we have in panel A, a Western blot of our control brains and Alzheimer's brains. And when we blot for total tau, we see that our whole cell lysate is the same, or similar levels. A fraction that has no ribosomes is about the same, but there's a dramatic difference in the accumulation of tau in the fraction that was only enriched with ribosomal proteins. You can see in the Alzheimer's brain, there is a robust accumulation of total tau, and much of this high molecular weight aggregated tau and insoluble corresponds to PHF1 pathological tau. Here, P0 and L28 are showing that our enrichment was successful. Here we have, these are ribosomal proteins that are enriched in these fractions. And HSP70, which is a cytoplasmic protein, was virtually undetectable in the ribosomal fraction as expected. So we further corroborated that in other pathological tau species. Here we have tau oligomers, which are recent evidence show that they're one of the more toxic tau conformers associated with this ribosomal protein, S6. Here we see overlap in the stain of tau oligomers in red with green, suggesting that the association is more abundant in a human Alzheimer's brain. So our first experiment in vitro was to test simply looking at tau and ribosomes and how they may, this interaction could affect translation. So for this, we did a cell-free assay where in different wells of a 96-well plate, we included all the machinery necessary for translation to occur. That is ribosomes, tRNA, initiation factors, elongation factors, et cetera. And we also included plasmids for GFP. So in different wells, we could incubate different proteins such as tau or controls, BSA in this case, and we can measure fluorescence over the course of 6 hours, which would be a surrogate of translation. Here are the results. In our BSA, bovine serum albumin control wells, we see that GFP levels increased over the course of 6 hours. But when we added tau oligomers, recombinant protein, the tau oligomers done the same, we saw about a 40% reduction in the amount of tau right at the end. And you can see that the curve is flatter in those wells that had tau. To determine whether this was the result of tau oligomers or specifically or other oligomeric conformers, we repeated the experiment using either alpha-synuclein oligomers or insulin oligomers. And we saw that compared to BSA, there was no significant difference in the GFP fluorescence over the course of 6 hours, whether the wells had synuclein or insulin oligomers. These data suggest that tau, that this effect in reducing GFP in translation was a result more specifically of tau rather than other oligomeric conformations. Our second in vitro model was using this inducible HEK line. Here we're showing that at 0 hours, there's hardly any tau, but once we add tetracycline, we see tau accumulating over the course of 4 days quite robustly. And we paired this model with a technique called sunset or surface sensing of translation. This is a very clever technique that takes advantage of the fact that pyromycin, an antibiotic, shares structural properties with tRNA. So for this experiment, we add pyromycin to the media on our cells, and essentially, and really speaking, pyromycin fools ribosomes into thinking that it's tRNA. And so new proteins now have a pyromycin tag, And with anti-puromycin antibodies, we can now detect not just the location, but also the amount of newly synthesized proteins without having to use radioactivity, which is typically what's done. So here are the results of our experiment using our HEK line. You can see that after inducing tau expression for 3 days, 24, 48, and 72 hours, there's an accumulation of total tau and certainly an accumulation of, again, PHF1 tau. Puromycin, which is measuring the amount of new proteins, significantly decreased as pathological tau increased, suggesting that there's showing an inversely proportional co-expression of these phenomena, both accumulation of tau and reduction in new proteins. But to determine whether this was more specific, we did a rescue experiment. And here we have our HEK cells expressing tau for 24 hours up to 96 hours. And you can see that there's the expected accumulation of PHF1 tau. But then after 4 days, we wash the media for 1 day or 4 days. So now we don't have tetracycline, and you can see that tau levels go back to baseline after 4 days of not having tetracycline in the media. And puromycin levels obey that same property that we saw here. That is, as pathological tau levels increase, puromycin decreases. And when we wash off tau, puromycin or new proteins expression and synthesis comes back to normal or even overcompensates a little. Finally, our third model was using primary neurons of these tau transgenic mice. We obtained these mice from Dr. Jada Lewis, who's actually down the hall from me. And these mice overexpress a highly toxic form of tau that has a P301L mutation that's associated with frontotemporal dementia. Now, these mice have, just as a caveat, recent data show that they, because of the random insertion of the transgenes, they affected expression of other genes. Nonetheless, in our primary neurons, they serve a great purpose, which is to establish whether or not translation of synaptic proteins is different. So here I'm showing a puromycin assay in primary neurons of our transgenic mice or controls. Here I'm showing that puromycin levels are also decreased, as we saw in our inducible HEK cells. But this synaptic protein, PSD95, also follows the same reduction that we see in puromycin. Here, H-tau shows that human tau is expressed only in the transgenic cells. However, to this point, we are not accounting for differences in transcription. So to this end, and having at least one protein that's changed, we did RT-PCR for PSD95, and we saw that compared to controls, which are here, RNA, oh, sorry, this is a quantification of the Western blot. Here's the RT-PCR. We see that in the transgenics, there is a highly significant amount, about 2.5-fold of RNA coding for PSD95. So to clarify this point, there's much more RNA for PSD95, and there's a lot less protein. These data suggest that there is something interrupting the conversion of that RNA into new protein. And it stands to reason with everything else that I've shown you to this point that tau impairs translation. And some of the proteins that are not synthesized because of this impairment certainly are important for synaptic function. So at least in vitro, our hypothesis that pathological tau reduces translation holds true. However, we weren't sure whether this would hold in vivo. So to this end, we went back to our RTG4510 tau transgenic mice, and astutely, the authors who co-invented this model had included a tetracycline off system. That is to say, when the mice are fed doxycycline or tetracycline in the diet, tau expression is suppressed. And this is a beautiful experiment done by Shelby Meyer, who was a graduate student in my lab in Kentucky, where she injected puromycin intraperitoneally in the mice. Puromycin went into the brain. And here we're showing in the darker color puromycin signal, which corresponds once again to new proteins. So here we have 5-month non-transgenic and transgenic mice. And here we have 7-month-old non-transgenic and transgenic. And while there is a decrease in puromycin as early as 5 months, this decrease was not significantly different, but it was certainly significant and pronounced at 7 months. Now, this is a bit perplexing because at 5 months, these mice already have substantial pathology and cognitive deficits. So it meant that either this decrease in translation was not driving cognitive deficits, at least not early on, but maybe there wasn't just a suppression. Maybe there's a shift in the types of transcripts that are turned into protein. So we hypothesized that, again, pathological tau impaired translation of proteins that were necessary for health, but it also shifted to make proteins that could help adapt to the stress or disease condition. So to this end, we had our tau transgenic mice. We looked at them right before 5 months because we wanted to look at the brains at a time point before there is significant neuronal loss. And by this stage, they already have significant tau expression, tau pathology, cognitive impairment, and deficits in plasticity. And as a control, we treated mice with doxycycline to make sure that they're expressing tau, that they have early pathology. And by suppressing tau expression from 3.5 to 4.5 during that month period, the mice actually recovered. This was published in 2005 in the Science paper. So here's just to show that when we treat the mice with doxycycline, there's about a 50% reduction in the amount of tau that's made, and they already show benefits or rescue in the function, rather. So let me show you that after the treatment with tetracycline or vehicle, half the brain was dedicated to do microarray to measure changes in RNA, and the other half, we did a pure myosin immunoprecipitation and tandem mass spec to identify the proteins that are translated or the translatum at that particular time. Looking at the RNA only, here we have our microarray results, and we found 3 distinct patterns. Here we have our non-transgenics, whether treated with vehicle or doxycycline, in our transgenic mice treated with vehicle or doxycycline. You can see that expression of these genes obeyed were rescued by treatment with doxycycline or that reduction in tau. Another pattern were these genes which were resistant to that reduction in tau expression by doxycycline. Our third was, and this is a very important control in any of these experiments, and that's those were responsive only to doxycycline. We are treating the mice with a heavy dose of an antibiotic, and so it makes sense to look at changes in the levels of any kind of genes and account for them when we treat with an antibiotic. But since we're interested in factors involved in translation, when we looked at those, whether it were ribosomal proteins or initiation elongation factors, these were not changed. Transcription of genes involved in the production of ribosomes and necessary for ribosome function were not changed because of tau, whether treated with a vehicle or doxycycline. So that way we know that any changes that we might see at the proteomic level were the result of translation, not of transcription. And here are some of the results. Again, we pulled down pure myosin from the brains of these mice and did our liquid chromatography mass spec. And we found that whether the non-transgenic mice were treated with doxycycline or not, there were no difference in the types of proteins that were made. And one group that really caught our attention were those that are involved in translation or ribosomal, because when we compared those transgenic mice that overexpressed tau, there was a massive suppression in the production of these genes, of these proteins. And when we treated doxycycline, they were completely rescued, suggesting that, again, in vivo, there is a change in the types of proteins that are made, and those involved in translation are reduced. So to hint at mechanism, we went back to some of these ribosomal proteins because we do see the selective mechanism by which tau changes the types of RNAs that are translated. And one of those proteins that caught our eye is this ribosomal protein S6 or RPS6, which is a major regulator of translation. And it caught our attention because when it's phosphorylated, it shifts the specificity of transcripts that will be translated. For example, phosphorylated S6 promotes the translation of proteins involved in cell growth. These are called 5' top mRNAs. And they have this motif, this 5' terminal oligopyrimidine motif that is present in all ribosomal proteins and in all initiation and some elongation factors. And here I'm showing data indicating that in our HEK cells, pardon me, in HEK cells expressing tau, here we see that there is a relationship between the levels of tau and S6. So for example, when we add tetracycline to the cells, after 1 day, we see accumulation of both total tau and PHF1 tau as you expected. After 4 days, there is a robust accumulation, and washing off tetracycline once again reduced tau levels. Phospho S6 levels were essentially undetectable after 4 days of tau expression, suggesting that there is this interaction between tau. So as tau levels increase, phospho S6 decreases. And therefore, we would expect a reduction in the production of proteins involved, these 5' top mRNAs involved in translation. Some of our early work, and this was a complete coincidence, and I showed you this data before, the tau oligomer is associated with this ribosomal protein S6. So then we did co-immunoprecipitation Western blot assays in human brains, and we found that whether we immunoprecipitated S6 or tau in Alzheimer's, in control brains, we saw an accumulation of the reciprocal protein and a co-immunoprecipitation, and that association was more robust in the Alzheimer's brains. So finally, and once again, controlling for transcription, we did RT-PCR of a handful of these 5' top mRNAs, RPS6 being one of them, EIF3E, RPL28, and EIF2S1. And in late stages of Alzheimer's, we see an accumulation. There's an increase in the RNA coding for these proteins. And this was from blot, I'm showing you that there's a significant decrease in the protein levels of all of these. Certainly, phospho S6 levels will also be reduced as it matches with the in vitro experiment. Once again, RNA levels are up, protein levels are down, suggesting that there's an impairment in translation. Some of the current work we're doing to follow up on this part is to establish some of the association. Where do these ribosome proteins and tau associate? Here I'm showing some early data indicating that the proline-rich region of tau is essential for the interaction. Here we've deleted the proline-rich region in tau, and we see that the immunoprecipitation of these two ribosomal proteins, S6 and L19, is gone. So RPS6 and RPL19, representative of ribosomal proteins and perhaps the entire ribosome, no longer co-immunoprecipitate with tau when it's missing the full proline-rich region or two independent proline-rich regions or domains within that proline-rich region. We've also, it's important to explore the association of tau with RNA. And here we're showing some data of tau. And given that certainly RNA has been established to form stress granules with tau, and this is work that Ben Wollesen's lab has done, and he's been very kind to allow us to come along for the ride and help out in some of this work. Ben has shown that some proteins involved in RNA stability and metabolism are critical for driving this pathological effect. So for example, HNRNP A2B1, which modifies tau, RNA, associates with tau and therefore may impair the translation and maturation as a basic fundamental biological process. We've done some of this work and we started by identifying all the RNAs that associate with tau in Alzheimer's brains. For this, we did enhanced CLIP or cross-link immunoprecipitation, which is basically focusing on co-immunoprecipitating tau from human Alzheimer's brains and doing RNA-seq to identify those transcripts that associate with tau. We had 3 different groups based on severity, mild or early, moderate Alzheimer's, and severe Alzheimer's disease. You can see that when we look at RNA levels, there was much more RNA that associated with tau compared to controls in Alzheimer's brains. The types of RNAs were very different as disease progressed. So for example, RNA that associated with the coding sequence was reduced as Alzheimer's disease progressed, but other groups, such as this distal intron, increased in the severe cases of Alzheimer's disease. We're not quite sure what that means. But when we looked at the consensus sequences that were identified in the RNAs that associated with tau, we found many interesting trends. For example, there seem to be regions that have poly A tails that associate with tau early on, and some splice sites. When we went down to the identity of the RNAs that associate with tau in Alzheimer's disease brains, we found that many of them are involved in RNA processing. Some of them went up in red, and some of them went down in blue when they associated with tau. Some of these that we're following up on, and Christian Lessard in my lab is doing most of that work, is focused on small nuclear RNAs, or SNORs. So to the final stretch of the talk, I've shown you that in these models of Alzheimer's disease and in human brains, we see not only a reduction of proteins involved in normal translation, like ribosomal proteins and synaptic functions, such as PST95, but there might be also an increase in other proteins that are probably trying to adapt. But chronically, overexpression of these adaptive proteins can lead to problems. And one of those is PERK, and I'll get into that. So PERK is an ER stress protein. And some of the early work that we did in 2013 is represented here in this schematic. Here we have a cell and the endoplasmic reticulum with ribosomes and PERK as a transmembrane protein. The ER provides an optimal environment for proteins to fold properly. Here's an example. This cylinder I've represented as a membrane receptor. And since it folded properly, it can go to where it needs to be and do its function. But not every protein will fold properly. And so here I have this ribbon that has been identified as not being able to fold in its proper conformation. So it will be translocated to the outside of the membrane, ubiquitinated, and destroyed in a process called ER-associated degradation, by which proteins such as VSP or VCP or P97 pick up that ubiquitinated protein and target it for degradation. What we found again is that tau blocks ERAD, and in doing so leads to an accumulation of ubiquitinated proteins surrounding the endoplasmic reticulum. This induces ER stress. And one of the first things that happens under ER stress is activation of the unfolded protein response in order to adapt the cell. One of the effectors of the unfolded protein is PERK. PERK dimerizes autophosphorylates, and its job is to reduce translation. Everything that we've said is that while tau is reducing translation, well, here this is important because it reduces the input of new proteins inside of the ER to give the ER some time to clear everything that's on the inside, destroy it, try to refold it, and come back to normal. That is an adaptive response. This is kind of how the unfolded protein response works. It early on has an adaptive pathway. Everything here in green is telling us, hey, we're suppressing translation in order to reduce input and clearing out things and promote survival, expression of survival genes. But imagine a neuron that can't make synaptic proteins for very long. Chronic expression of this mechanism eventually will shift and promote the expression of pro-apoptotic proteins. It's almost as if this cell was unable to cope with the stress event and therefore needs to be targeted for cell death. PERK became very interesting to us because it is genetically, pathologically, and as we found, molecularly associated with tauopathies. That is to say, there are SNPs on the gene coding for PERK that are associated with this tauopathy, PSP. There are phospho-PERK, active form of PERK aggregates inside cells in these disorders. And as we found molecularly, tau activates PERK. What we found, so part of the mechanism that we thought was that chronic expression, as I said, will activate pro-apoptotic proteins. And this is evident in different models. We've done some of this in TBI and spinal cord injury. This is preliminary data. This is the work that we published in 2013 and some other work that we followed up with collaborators in the Sapienza University of Rome, looking at Down syndrome. But the hypothesis was, if pathological tau species indeed induces ER stress and activates PERK, which in turn leads to long-term reduction of translation, then we could block PERK and rescue translation. We use this, we used a PERK inhibitor that was given to us by GlaxoSmithKline, so I wanted to disclose that as a conflict of interest, that this compound inhibits PERK. And so here it was first developed as a PERK inhibitor, but we found later that a high concentration of 414 can lead to a broad kinase inhibition. So this is just kind of a heads up on what you're going to see. 414 indeed rescues translation. Here we have tau transgenic mice treated with the drug for 2 days or 4 days, and you can see puromycin levels in 8-month-old 4510 mice are reduced. And as soon as we treat the mice with the drug for 4 days, puromycin levels come back to substantial and significant amount. So the 414 PERK inhibition strategy increases translation, which is exactly what we want. Our treatment strategy was, again, to treat mice right before there is neuronal loss, and somewhat of a preventative treatment. That way we wouldn't have overt PERK activation as we had described in 2013. One of the first observations at 5 months doing MRI was that after 1 month of treatment with the PERK inhibitor, we saw that brain atrophy, which is evident in these mice, treated with transgenic mice treated with a vehicle, was protected by the compound in those mice treated with 414. We then did this manganese enhanced MRI as a functional assay using manganese. Because it enters voltage gated calcium channels, it is similar in size and identical in charge to calcium. So it also is a surrogate of calcium, and because it's paramagnetic, it can tell us with MRI in the T1 or R1 relaxation time where the manganese is. And since calcium is critical for neurotransmitter release, it tells us a little bit about function. This is more or less what it looks like at baseline. You see mostly yellow and blue, which is the absence of manganese. And after 6 hours of injection with manganese chloride, manganese spreads throughout the brain. And here are the data. In our normal mice, we see that the R1 relaxation was about the same. There are no significant differences in the transgenics, whether treated with vehicle or not, although there's this decrease in the non-transgenic treated with the drug. This is what the transgenic mouse treated with vehicle looks like. Structurally, look at how thin that hippocampus is. And in fact, there's this hole. That's where CSF is filled in because of the gross atrophy that this brain undergoes after 5 months. But the transgenic mouse treated with the drug had protection. As I showed with the T2 analysis, here we see not only protection in structure, but also in function. The blue column in the dentate gyrus and the CA1 shows a rescue in the dynamics of manganese flow, representative of calcium flow and brain function. We also did other behavioral assays. For example, this is the open field test. These mice are characterized by having an anxiety-like phenotype. That is, that the mice run around in the periphery. And this red line traces where this mouse is running in this field. And as soon as we treat the mice with the compound, this particular mouse didn't seem to care very much about where the periphery or the center was. They didn't seem to have this anxiety-like phenotype. We also did Y-maze deficit measurements, and we see that there's a deficit in the recognition of the novel arm by the mice treated, the transgenic mice. But when they were treated with vehicle, we saw that they seemed to remember that new arm of the Y-maze. Mechanistically, the drug did not reduce tau levels. Here's a Western blot showing that, and our proteomics also shows that there was no significant change in the amount of tau. So this is interesting because the drug has an effect, a functional rescue that is independent of the accumulation of tau. And in my view, is very important because it allows us some kind of, say, hope that this treatment could confer benefits in a population that already has tau pathology. However, it's not that simple. As I mentioned, this compound at high enough dose, like the one we used, will certainly inhibit other kinases. And so we did some TMT proteomics to identify those kinases that could be affected by the compound. And we narrowed down almost 200, 186 proteins that were rescued. Many of these were kinases. These are just some examples of, to validate them, the ones that we expected. Tau again didn't change. But they're involved in important processes. So for example, again, here we have our pure myosin assay showing that there's an increase in translation in these mice. Here we're showing a marker of, say, protein scarring by nitroxidation. Treatment with 414 reduced the levels of the scarring or nitroxidation. Other processes that we would have expected to be changed based on those 186 proteins was lipid peroxidation, but it certainly did not, according to these results. 4H staining did not change lipid peroxidation. But as we narrowed down using different data sets from the Nick Seyfried group at Emory, able to narrow down to 4 proteins that seem to play a very important role in the rescue. And these are involved in, we're not sure what they're about. We're currently validating these. Paxin-1, there's only one paper of Paxin-1 with tau. There is, we were not able to find a clear association between these other 3 proteins and tau. So these studies are currently underway. And just to finish up, this is a summary of the work that I've presented. The tau associates with ER proteins, many of which participate in RNA translation. The pathological tau interferes with the ribosomal function. This phenomenon occurs in Alzheimer's disease, where tau engages with this particular and interesting protein S6. In vivo, it shifts both the transcriptome and also the translatome. We want to keep that in mind for future studies, even though we didn't explore it. Studies to identify the proteins altered by the tau ribosome complexes are currently underway, also to understand the tau RNA association and what are the implications of it. And finally, this study with the PERK inhibitor, more as a broad kinase tool, revealed new potential mechanisms that are now of interest for new strategic therapeutic strategies. So with that, I'll end the talk, thanking the lab. Certainly, the lab is currently composed by many graduate students, undergraduate students, postdocs, and senior faculty in the lab. Many collaborations essential for team science, both at the University of Florida, particularly Dr. Goldie and Dr. WashU. University of Kentucky, Dr. Peter Nelson, who gave us a lot of the human brains. Nick, who did a lot of the proteomics, our collaborators at Sapienza University of Rome, GlaxoSmithKline, and certainly Chad Dickey, who inspired most of this work. To begin with, our funding. And with that, I am happy to take any questions. Thank you very much.
Great, thank you so much. That was really great and really an amazing example of how powerful it is to work across systems and across groups. So I think if anyone wants to unmute and ask a question, please feel free. If you're more comfortable, you can type your question into the chat box and I can read it out.
Thank you, Joe.
So,
Yeah, can I ask
Sure.
a question?
Thank you.
Sorry, Celeste.
no, it's okay.
Yeah, I'm just so curious. So, why S6 out of all the interactome? Is there a specific reason? And do you think other ribosomal RBPs play an important role as well?
You know, this is one of those moments that I wish I'd had a clear rationale to say, well, this was obviously because of X, Y, or Z. But honestly, here's the reality of science in my perspective. I had sent out a mass email to colleagues in Kentucky and said, I was really excited we had these who has a ribosome protein that I can test today? Because I can't wait till tomorrow. And I went to the lab next door to John McCarthy's lab, and he said, we have this one. It's for S6 and phospho S6. Okay, sure. And we pulled it out, and certainly there were these changes, and did the human staining in 2016. So we already knew that tau associated with this protein. So, well, let's go with what we know in the 2019 work. And sure enough, there it was, that phospho S6 was there. But that's the exciting part. Now we can trace down all the others. And we have another paper in 2015 that lists about 12, I think, ribosome proteins that could also help us understand the interaction and now the motif and understanding where these ribosomal proteins associate with tau. So it was sheer luck. But you know what? They say that luck is a series of good decisions. So, so I don't know.
Good insights.
Great. So we have a question in the chat from Liz Bradstreet, who says, great talk. And what do you think about the idea of tau as an antimicrobial peptide?
Tau is an antimicrobial. I have no thoughts on that, Liz. I'm sorry. Would you tell me a little bit more why you're thinking this? What's this? Why are you inspired to ask that question?
Liz, you're welcome to unmute, but if you prefer not to, we can.
That's okay. Yeah, we can maybe typing.
I think one thing I was really.
Celeste, what do you think? Why don't you give us some insight? You're an expert in tauopathy as well.
Yeah, around the antimicrobial.
Yeah.
I don't know. I honestly, I'm not as well versed in that area, I think. But I certainly think that Liz doesn't have a microphone, so
Yeah,
we understand. I certainly think that there's an important intersection of the immune response in the brain to any of these neurodegenerative diseases, and understanding what can initiate those responses and what can also sort of break down the normal sort of reactivity and then reactivity and relaxing is important, and whether tau triggers those, I'm not sure. I also, oh, sorry, go ahead.
Yeah, go ahead.
I was just gonna say that, one thing that struck me about your work is that, with this overall increase in protein synthesis, I'm thinking about the impact of all of this extra RNA around and thinking immediately about sort of some of the non-coding RNA and all of the mischief it can get into, and how that can be quite sort of pleiotropic in their effects. And have you looked at some of the non-coding RNAs and their impact on downstream pathways that might be relevant to tauopathy?
Well, we've identified those that I'm very happy to that I shared on the table that were. I mean, we did some really rigorous cutoffs to highlight just a handful, but you could see in the slice of that pie there were hundreds of non-coding RNAs, and many of them don't have a defined function. So that tells us a lot of cool things. First of all, the promise that there might be something there that's very interesting. Another one that maybe is just completely stochastic because there's an abundance of it. But there are those small nucleolar RNAs, and that's what we're following at this time. And what's cool is that the small nucleolar RNAs work in tandem with HNRNPs, and the SNORs and the HNRNPs seem to work together. And so Ben Willowson did that work with HNRNP and got that paper in Molecular Cell. And so we're following up on that other side that has a different function on modifying RNA for maturation. So there's a lot there, Celeste. There's a lot. And it could very well be, and honestly, one of the things that got me interested in these dynamics of tau and aggregation is, as you know, you make recombinant tau, put it in a tube, and it won't aggregate for months. It's very hydrophilic. But then you add something like heparin or arachidonic acid, and it facilitates aggregation. Well, it turns out that polyanionic structures, such as RNA, also accelerate tau aggregation. So if there's an accumulation of RNA in the cell, the question is, does that facilitate the formation of stress granules?
Great. So we have another question in the chat from Eunice, asking whether pathologic tau may play a role in regulating translation and ribosomes under some condition.
Does pathological tau play a role in regulating translation and ribosomes under some condition? Yeah, so that's the idea. Maybe what's happening is that in early stages, tau released from the microtubule associates with ribosomes, and that suppresses ribosomal function because the cell is perhaps trying to rescue its normal function or get rid of whatever stress event is there. But long-term association of tau with ribosomes and RNA would have negative consequences. So maybe this is an early stress response, and if it's not mitigated, it would lead to long-term consequences, dire consequences, and promote cell death or dysfunction, worse yet. I don't know if I answered your question, Eunice. Oh, yeah. So that's what I think. It's the same strategy of, it might be a stress sensor, recognizes a stress response and it has an effect, but long-term it is detrimental. And, imagine a head injury that promotes ER stress or tau release to induce that ER stress to suppress translation. And 20 years later, that accumulation and that effect that cannot be resolved leads to pathology. It's 20 years of an abnormal mechanism that can't be stopped. Just speculating. You're welcome.
Great. I think we have time for one last question if you all have any additional questions.
If not, I have a question for you, Celeste.
Okay.
How,
on.
so as you know, Matt Lavoie is down here, and we're interested in, and I'm interested in looking at other models, maybe iPSCs. Do you think some of this is viable in iPSCs, given the challenges in growing iPSCs and differentiating them and maintaining in some line? I know that Matt uses mostly I neurons already partially differentiated. Also the question of cell type, right? We know PS95 is affected, suggesting that maybe neurons are the ones that are affected. How about in dysregulation of ribosomes in microglia? We're talking about inflammation.
Mm-hmm. Yeah, I mean, I think practically it's possible to do these experiments. I think one question that I would have is in the iPS-derived neurons. One question I would have is, what are some of the critical initiating factors? Is this something that's happening so early and really driven by the mutation alone, or is it some sort of intersection of the mutation plus aging? In which case it might be, you might need to stress the cells in some way or accelerate aging in the dish in some way. Because in general, in our tau mutation lines from patients who carry these different mutations, the neurons don't generate huge, robust, massive aggregates. And we think that's due to a few different reasons, but in part due to them being relatively young still in their chronologic age. So I think that would be the biggest question is, do you need to give a second insult beyond the mutation to see an effect?
How about, are there iPSCs where you can, I mean, as far as I understand, they don't make 4R tau.
They produce very low levels of 4R tau.
Okay.
Yeah.
Okay.
We can increase 4R tau with mutations in the MAPT gene that cause FTD, like IVS 10+16. And there's a new paper that I think is out from Andrew Yu's lab in stem cell showing that directly converted fibroblasts, so fibroblasts that go directly into a neuron, maintain the 4R expression of tau.
Okay, cool. All right. Well, maybe we can talk offline about some of these things that we could do.
Yeah.
Pretty fun.
That's exciting.
Great. Well, thank you all for joining us. This has been really, really fantastic.
Thank you
very
so
much,
Joe.
Thanks for the opportunity. This was a lot of fun.